Welcome. My name is Jeff Krause. We're going to do a little bit of a tag team seminar, which we have lovingly called our PhySSi World. And I want to introduce the members of my lab that are going to assist. Includes Lacey Bowman, my technician. She has a BS in marine biology from the University of West Florida, and also an MS in biology from the University of West Florida. Priya Gilman, who is a PhD student that's co-advised by Brandy Keel-Reese and myself. She has a BS in marine science and chemistry from the University of Alabama. And what I just remembered today from reading her ZV, minors in interdisciplinary environmental science and Spanish, which is pretty awesome. And then Antonia Thielecke, who is a student who I'm on her committee. She has a BS in biology from the University of Freiburg, and she did her master's at University of St. Andrews in marine ecosystem management. And she is joining us from the Alfred Wagner Institute in Germany. I'm Jeff. I've been here since 2012, and I'm a senior marine scientist and a professor at the University of South Alabama. And this is some of the places we've worked, and especially considering the number of people that we have kind of new to the lab, we felt this was a good way to introduce the type of science that we're doing, some of the questions, and then hopefully if you guys feel inclined, you can actually come and talk to us, because it is really important. And a fraction of what we're going to show you today, all the sediment stuff, actually grew out of a conversation with a PhD student in Dr. Carmichael's lab. We had a conversation in 2013, and that has now become a really strong facet, that sediment facet of what I do. All kind of, I can trace it all back to Beth Condon coming into my lab, sitting down and talking about how to analyze sediments. And so be open to ideas about, talking with your peers, because they can really transform your trajectory. So let's get going. Of course. This is resume slideshow. How about that? Here we are. Okay, so the big question is, what is a PhySSi lab? Well, it's an acronym, like many things. It stands for phytoplankton, sediment, and silicon. Formerly, that first S was for sulfur, but we have kind of rebranded. So we were established in 2013 when the first members of my lab started, and we rebranded twice, mainly because we expanded. So in 2019, after the passing of Ron Keene, we brought all 4 of his students into our space, and we renamed ourselves as a nice team building activity, and it just kind of stuck. We did this activity again because we liked it so much when Sean Wang's students came to my lab, and also to Dr. Carmichael's lab. And then we've had this tremendous turnover in the last year, where 5 students were out the door last year, and we have 3 new students in. And so we have yet rebranded. So it's pretty exciting. So who funds our work? This is always important, and it's kind of an interesting thing, because when a lot of people ask what we do, I think you might get the same questions like, oh, the university just pays for all of this. And you guys know that's not true. We need to get external funds. And but we do want to kind of acknowledge that, the people that pay our check are very important. So I'm an employee of the Dauphin Island Sea Lab, and my students are employees of the University of South Alabama. So thank you to all of them. But largely, you guys, if you ever pay taxes, you guys pay for a lot of the research that we do in my lab. This includes most projects from the National Science Foundation. Well, National Science Foundation is just one of the organizations that funds basic research. Another organization within the federal government is the North Pacific Research Board, mainly set up to do research to kind of preserve the resources in that region. Think fish. Food and Drug Administration. We also have ALCO funds, which is through the state of Alabama and also the Department of Treasury. And then we have private entities such as the former Gulf of Mexico Research Initiative. And then we have some international funding that we've gotten over the years. So where do we do research? Well, we do it in a lot of places, or we have done it in a lot of places. I've never been one to be kind of tied to one thing, which is why I really like this microphone, and allows me to migrate and move. And you can think of my research doing the same thing, because I go to where the questions are. And if you know anything about diatoms, those questions usually take you to the poles. So we've worked in all these different regions, and currently we are working at both poles. And some of our members right now are with the rest of Dr. Keel-Reese's crew in the Ross Sea. So this is us. And you can see the individuals that I have just introduced, Lacey, Priya, and Antonia. And you can see the rest of us, and then also, our alumni. In fact, the top two here have kind of stuck around in the C-Lab family. Kyle is at DHP and Blair is at Mobile NEP. So this is who we are. This is where we get our money. What do we do? So we're silicon biogeochemists, right? We think about phytoplankton, we think about geochemistry, we think about sediment. So why do this? What's the importance? So I can give you a primer. I'm going to put the bio in biogeochemistry. If you think about the kind of the main four phytoplankton groups in the ocean, you have cyanobacteria, haptophytes, dinoflagellates, and diatoms. All of them take inorganic nutrients. Through the process of primary production, they turn that into organic matter. But diatoms have this kind of special path. They have this special path because they have a requirement for silicon. It's an obligate requirement, meaning they have to have it, otherwise they cannot grow. And because of that, they use it to build these cool shells, which we call frustrals, and they make this frustral out of biogenic silica. That's a term you're going to hear a lot today, biogenic silica. Now, these guys alone, well, that means that these are all essential elements for diatoms. Now, because diatoms are so prolific, in fact, they give us about 10 to 20% of global oxygen production. That's about the same quantity of all the rainforests combined. Because of that, there's this really strong coupling between the biotic cycling of silicon with the cycles of carbon, nitrogen, and phosphorus. Now, phytoplankton, even in general, we know that they're the base of the food web, but another thing that you might not think about is what happens in the sediments to this organic matter that they produce, because that's the basis for a lot of fossil fuels. And diatoms have been really important with this in the last 60 million years. So that's the bio, but the geo in the geochem, in the silicon biogeochemistry, it links to this, but it gets a little bit crazier. So there's two processes that kind of regulate climate in the ocean that have to do with the silicon. If you're thinking about geology and silicon weathering, basically when other rocks get weathered, that liberates silicon into rivers, which makes its way into the ocean, and that actually consumes CO2, and it produces these kind of like degraded aluminosilicate phases. Now, if you go downstream and into the ocean, you can actually reverse that process, which we call reverse weathering. It's not a true kind of surrogate or a true backwards reaction, but you should take our diatom organic matter. You can either dissolve it or you can have things reprecipitate to it. And those reprecipitated things are aluminosilicate clays. What's important about this is it gobbles up all those elements that are in the clay and it produces CO2. So if you go a little bit bigger picture, you can look at the distribution of silicon in the oceans. You can see that, in most of the ocean, it's pretty low, and at the poles, it's pretty high. And then if you look at silicon in the sediments, you can see how we have, really high silicon concentrations that are in the pore waters of sediments. And there's a lot of weirdness that happens here. This is what the pore water should be based on the solubility, and this is what the pore waters are. And so just looking at these are kind of the bookends of what we do research-wise. We can tell you some main questions that my lab aims to answer. First, how does silicon affect the contribution of diatoms to primary productivity? This has really important implications for things that I'll talk about after this animation. And in the sediments, which processes sustain this undersaturation of silicon in global sediments? Okay, so these are big questions, and what do they mean? Well, they mean a lot because diatoms are usually the basis of many marine fisheries. If you go into a big fishery, think the Menhaden in the Gulf, think walleye pollock in Alaska, think anything in Monterey Bay, these are diatom soup, right? These are places where diatoms are exceptionally abundant. And the reason why they're abundant is because of the energy in the systems. And what that does is it makes shorter food chains. And that is a more efficient food web, which allows you to funnel a lot of biomass to upper trophic levels like fish. But it also means something for CO2 in the atmosphere, because as that organic matter sinks, because diatoms have a shell made of glass, that moves CO2 from the atmosphere down to the deep ocean and helps regulate the climate. In the sediments, if you sequester the silicon in the sediments, then that silicon doesn't get back into the water to feed the diatoms. But if you release it, it can. Now, also sequestration, as you saw on that first kind of slide with reverse weathering, that consumes silicon and it produces CO2. So diatom silica it's really important kind of at both ends of the spectrum for regulating CO2 in our ocean and atmosphere. So I'm going to pass this off to Lacey, and we're going to tag team a little bit, tell you a little bit about what we do at sea, where we've gone, and we'll even present some data.
All right, thank you, Jeff, and hello, everybody. So as Jeff said, I'm going to kind of go through where the PhySSi Lab has been. So we've been through a lot of places. The members of the PhySSi Lab have participated in up to 33 cruises on 20 different projects, give or take. And those cruises range anywhere from 1 to 44 days. And we've been to lots and lots of different places, kind of pointed out on our spill house projection of the world's oceans. So 2 cruises that I'm going to talk about in particular are 2 that I participated in. One is going to be the qualified cruises. That was off the Louisiana shelf, and this happened last September. And also the Snowbirds 2 cruise. And this one was in the Southern Ocean along parallel 170 West last fall as well. So let's get into it. So I'm going to go into a day-to-day of the qualified cruise. We did lots and lots of different things, not limited to, but including CTD profiles, hydrocast. We did some sediment core sectioning, and we also did some sediment slurry incubations and lots and lots and lots of shipboard analyses, mainly lots and lots of dissolved silica analysis. And you can see over to the side, we've got a time lapse of just 1 day's worth of sediment core sectioning, probably starting around 7 or 8 and ending just before lunch. Now, on the Snowbirds cruise, we also did a whole lot. This was a cruise I spent 44 days on. The qualified cruise was only an 8-day cruise. And we did, again, some more CTDs and hydrocast. We did some opportunistic molecular sampling of some sea ice, pancake ice. We also did some McLean pumps, diatom assemblages, Poulter experiments, and vertical net tows. Lots and lots and lots of stuff. And lots of stuff not pictured here. In addition to all of the field sampling we did a lot of shipboard analysis, including chlorophyll A, dissolved silica, nitrate, and a whole other slew of shipboard analysis. So we do a lot of stuff on these cruises, but we are always limited to the weather. And anyone who's been on a boat for an extended period of time knows you got to work with it. Both the really nice, beautiful, pretty days where surprisingly the Gulf of Mexico is like glass. But on the other hand, you also have to deal with the bad weather days. So, 20 and 30-foot seas in the Southern Ocean, or a hurricane that comes right along your sample site off the shelf of Louisiana. So hurricane, the tropical storm Francine, which I believe was a hurricane when it made landfall, came right up through our sampling site 2 days before we left for qualified. So we actually had that cruise cut short by that hurricane. But it was really nice. We still got to go out and do 8 days worth of sampling there. Now, despite all the hardships and dealing with things, we get some great rewards out of going out on these cruises. And I'm going to go through some of the shipboard data that we produce on the Snowbirds cruise. So this first plot is showing across latitudes and across down depth, both the temperature of the water and salinity. So the color is the temperature and the lines near the top are salinity. And so as we move from the south and these higher latitudes to the north, and as latitudes get lower, you can see kind of these distinct bodies of water. So very far south, we've got really, really cold, sub-freezing surface temperatures that get a little bit slightly warmer as you go down. And those water temperatures increase at the surface and at depth as we move north. Now, these temperature profiles give a really good indication of certain zones that we find in the Antarctic. First being our Antarctic zone here, kind of characterized by that really cold sub-freezing surf water that gets a little bit warmer as you go down. And then the polar frontal zone, which is kind of the transition zone between the Antarctic zone and the sub-Antarctic, and kind of the barrier or the boundary of the Southern Ocean. And you can see those slightly warmer subsurface temperatures, and actually a little bit of that warmer subsurface water is kind of coming into the Antarctic zone, right around in the middle right up here. So really cool little temperature profile we got from our CTD data. Now, the next thing I'm going to go into are some of the shipboard nutrient data that we got from the Snowbirds cruise. And on the top, we've got our dissolved silica. And the bottom, we've got our shipboard nitrate that we got. And what's really cool is you can kind of see these differences between both our Antarctic zone and our polar frontal zone. So if you don't know anything about the Antarctic zone, and Jeff kind of alluded to this a little bit, we've got really, really high dissolved silica concentrations near the poles. So further south in the higher latitudes, you can see really high concentrations of dissolved silica that increase with depth. And as we move north at the surface, you can see those dissolved silica concentrations going down. And that indicates kind of the presence of a bloom. As we move north on this cruise, we saw bloom-like conditions, so increases in fluorescence, and also drawdown of these important nutrients for diatoms like silica and nitrate. And so that brings us back to Jeff.
Right. So Lacey talked about how we kind of geo-contextualize a lot of our transect sampling when we're out at sea. And she talked about some of the stuff that we can do at sea, but there's a lot of stuff that we do which we cannot run at sea. And anything that has to be analyzed back at shore in a lab is pretty much everything, right? For a lot of what we do. So what we're doing at sea gives us a snapshot of what we think is going to be going on in the process. But then a lot of the real data, the stuff that we write the papers about, that's what is created back in the lab. And so we use a lot of tracers to understand processes and how diatoms are operating and affecting things in the ocean. So tracers, specifically stable isotope tracers or radioisotope tracers. So we're going to talk a little bit about stable isotope tracer. So stable isotopes are very useful. You know, I know many labs here use stable isotopes of carbon, stable isotopes of nitrogen. These are very useful. Carbon can trace like organic matter source, whether it's like a Spartina versus a Juncus marsh. They have very, very different carbon signatures. Nitrogen is exceptional for trying to help you understand your position in the trophic web. So you could look at, you know, the nitrogen content of fish tissue to figure out if a fish is feeding, you know, where they're feeding in the food web. We can take those same ideas and we can use them in the silicon world. And the isotopic composition acts almost like a fingerprint. So the stable isotopes of silicon that we use, we have three. Silicon 28 is the most abundant, silicon 29 and silicon 30. And a lot of times what we're doing is we're looking at this heaviest isotope, silicon 30, to give us a sense of what's going on biochemically. Like carbon and nitrogen, we have an international standard. This is called NBS 28. It's basically quartz, and we analyze that, and then we analyze our samples in reference to that. And so what happens over time is, like these other biochemical processes, the heavier isotope is just a little bit harder to use because it is heavier. And so if you have a diatom, this is a paper from Krause. She's a former postdoc at UC Santa Barbara. But if you have silicon 28 and 30, they're gonna gobble up that 28 first because it's lighter. And then they're going to kind of begrudgingly start to use that heavier stuff because they don't really have a choice. And then by the end of the incubation, when the heavier stuff represents pretty much all of it, they're just going to use it. Does anyone like pistachios? I love pistachios. And when you're in the bag and you pull that first pistachio out, you love it. It's great. And then you get that pistachio that's not quite cracked. So what do? Well, you can't waste it. So you throw it back in the bag. And at the beginning of that pistachio bag, you got maybe 1% of your pistachios are the uncracked nuts. But at the end, you're left with all of those things being uncracked, right? You have fractionated against that tough to crack pistachio. This is basically what diatoms are doing with silicon. But diatoms aren't the only thing that do that. Chemical reactions just naturally do this. So when you think about your silicon 30, just think about the pistachios in your bag that you threw out or didn't throw out. So this leads us to using this type of kind of idea and this type of tool to understand what's going on in sediments. And we, as Lacey said, this is a nice segue because we have been lucky and unlucky with hurricanes locally. And this is Hurricane Ida, a monster of a storm. Category 4 hurricane, exceptionally devastating. It laid waste to the Louisiana shelf. And, you know, it feels kind of disingenuous to say, oh gosh, I want to go out and sample after it because of the damage it did. But we actually did get that opportunity to go out, and it is a storm event that doesn't hit land very often. And we happened to be out about a week before it occurred. And then this other cruise that Lacey talked about that we ended up getting out to see 2 days after the storm passed. So we've been lucky to understand and be in a position to understand hurricane effects. So one of the stations that I'm going to show you, we enthusiastically call it Station 1 because it was the first station. And we were able to sample it, you know, 5 different times between August of 2021. Hurricane Ida hit in the end of August, early September. And then we went out 4 different times. You know, processes in the sediments, you know, kind of take a little bit longer to, you know, rebound than they do in the water column, which is constantly moving. But this gave us an opportunity to think about what's going on. And so we used these tools, and we use these tools that we have at our disposal in the lab combined with some standard methods. So Lacey showed you that method of nutrient analysis. Well, a lot of what we do in the lab is we have what we have, and we have what we can measure. And we turn what we have into what we can measure. And so we have a particle of silicon in a sediment. We turn it into something dissolved that we can measure. We can take silicon and we can leach it with a sequential extraction of sodium carbonate. And then we can also think about those reverse weathering products, those coatings, and we can strip those off with acid, and then we can do that carbonate digestion. And when we do that, we notice that, you know, we get more silicon that's released when we take those coatings off. And isotopically, it's actually a heavier material. It's material that might be more diatom. And so what this tells us is that this difference between these 2 types of methods tells us how much this reverse weathering is having an effect on the population or the assemblage in the sediments. Mind you, this is assemblage, they're dead, so they're not growing. So before the hurricane, we went out, the material here in the black, this is basically that second method where we leach stuff off first, and we notice that it's a lot isotopically heavier than the stuff that's not leached first. And there's this big difference that we've seen it every time we go out there. But then we went out after the hurricane, and all of a sudden, that got wiped out. So what happened was we used to think about sediments that when this happens, that material, it's just kind of locked away. What this showed us is that a hurricane kind of reset the system. It took those coatings off. It changed things. And then what's even cooler is they grew back in a couple months. Now, why do I say that's cool? I'm going to tell you in a second. But the interesting thing is this stuff is all isotopically light relative to the dissolved silicon in solution. And if you know anything about kind of isotope chemistry, usually isotopes get, you know, heavier in the particulate phase. But this actually is getting heavier in the dissolved phase because of all these chemical reactions that are happening rapidly. So rapid that basically we saw them on the order of months. 30 years ago, people thought these chemical reactions were on the order of 10 to hundreds of years. And in places like the Southern Ocean, maybe 10,000 years. This is exceptional. We were able to visualize it with the tools that we have. So another tool that we have, which is going to be discussed by Antonia and Priya, is using stable or radioisotopes to actually probe for rates at which things are going. And so we can do this for biological production, or we can use it for like passive uptake. And so Antonia and Priya are going to talk about that. But basically, you take silicon or some sort of a tracer that's in seawater, you add it to either water or you add it to sediment. If you're incubating, ultimately your diatoms, if they're in the water column, are going to take that up. They're going to take that label. They're going to put it into their shell. Then you filter it and you quantify the radiation. If you're doing this kind of passively, we can basically look at how much is left in solution, and we can think about how like the bacterial assemblage has changed that. And so Antonia is going to kind of talk next about some data from the Arctic.
Thanks. Yeah, so Jeff has talked about a lot about silicon, and if you think about primary productivity in the Arctic and you ask the average person working in the Arctic what limits primary productivity in the Arctic, they go nitrate. Period. Silicon doesn't play a role. And actually, some of Jeff's previous work has shown that that is not entirely the case. But what is also true is that there's not a lot of data on silicon in the Arctic. So that's kind of most of the literature that I know about that is looking at silicon in that area. And for example, Jeff's work from Svalbard has shown that actually at the end of the spring bloom, silicon co-limitation plays a role in regulating diatom primary productivity. And so what I set out as part of my PhD is to go more into the Central Arctic and like try and fill in a little bit that blank space in the center and look at what the role of silicon might be up there. And so I joined a cruise in 2023 with the German research icebreaker Polarstern that kind of went all the way into the Central Arctic up to the North Pole. And what you see in the colors here are the silicic acid concentration, so the dissolved silicon in the water. And you see that kind of for the majority of that cruise, it's very, very low, which makes sense. It's late summer. Most of the nutrients are already taken up. And then we've got this one station that has higher silicic acid concentrations. And so what you kind of need to know about the system is that the Arctic is kind of to a certain extent split in two when talking about nutrient, especially silicic acid availability. And so there is the kind of more American Canadian part that has plenty of silicic acid because it's kind of coming in from the Pacific and it's coming in from the like shallow shelf seas. And then you have the more like Eurasian part over here that gets a little bit of silicic acid coming in through from Strait, coming in from the Atlantic, but most of it is like very low silicic acid. And so we don't know exactly where the high silicon concentrations here are coming from, but it's quite possible that they're kind of washed in from the kind of Canadian side of things. And if we compare our nutrient availability with our phytoplankton composition, then we actually see that at this one point over here, which is station 4, what we see is here this large peak in dark green, which is centric diatoms. And so this is cell counts, and all the blue stuff you see is teeny tiny flagellates, whereas all the diatoms are rather big things. So while this doesn't look like much, what we see at the station is that diatoms are definitely dominating the community in terms of biovolume. So looking at that, you might think, oh, okay, we've got high silicic acid and we've got diatoms, so maybe actually silicic acid is important in driving primary productivity and community composition. So I looked at that a little bit more mechanistically as well. And so I ran the tracer experiments that Jeff just mentioned, and I made them a little bit more complicated. So I had a gradient of 5 silicic acid concentrations, and then I also had a gradient of light because we're in the Central Arctic, everything is ice covered. Generally, people assume that everything is light limited. So I thought, okay, why not look at that? And then for every single one of my bottles, I added 3 different stable isotopes. So I added carbon, I added nitrate, and I added labeled silicon. And what that allows me to do is for every single one of these bottles, measure the uptake of those 3 nutrients and kind of compare how they might behave differently across these gradients. And for today, I'm going to focus on the silicon side of the story. And so what we see is kind of focusing on the left side first. What you see up here is kind of, this is the light gradient, and this is then the silicon uptake. And what you see is that there's not really a light effect. So they don't care how much light they have, they take up silicon or they don't. It's definitely not light dependent, but what we see quite nicely down here is that the more silicic acid you give them, the more silicic acid they're going to take up, which is all fine and dandy and makes a lot of sense. Doesn't quite answer the question yet whether they are actually silicon limited as they are doing that. And so what I did is I looked at relatively simple Michaelis-Menten kinetic, which is a way of describing the uptake of nutrients, and you kind of assume that at some point the diatoms have enough silicic acid and they stop taking up more as you give them more. So it kind of just saturates, and that's also what we see in this data here. And what you can do by calculating that is that you can use this plateau as like a maximum potential uptake rate, and then you can compare that maximum potential uptake rate to your in situ uptake rate that is actually possible with the silicon they have available in the system. And what you get out of that is kind of a number for limitation. And so that is what you get over here. And what has been shown in lab experiments is that diatoms are very good at reducing the necessity for silicon that they have. So kind of for a long time, as silicon goes down, they can just make slightly thinner shells and still keep growing. That is what we call kinetic limitation. But that at some point, silicon becomes so limiting that actually they need to reduce their growth rate. And lab experiments have shown that this kind of magic line is around 0.25. And what we see in our data is that here, the kind of stations that had low silicic acid are actually below this potential growth limitation. So what we can say with quite a bit of confidence now is that as long as they don't get random bursts of silicon, which might happen in the very central Arctic, across the kind of majority of the European Arctic, diatoms are in fact co-limited by silicic acid. And that's kind of the end of my little Arctic story, and I will hand over to Priya for some more local Louisiana stuff.
Yeah, so my project is about the microbial impact on authigenic clay formation. So we're looking at how microbial communities in the sediment might be accelerating or changing the rate of authigenic clay formation. So the first part of my project is field-based. So this is what I've been doing this past year. We have 3 research cruises, and we've been on 2 so far. These are some of our pictures for data collection. And we went on one in September, one in January, and then we will be going on one in May. And we are doing some observational studies where we are taking sections of sediment cores and comparing basically the microbial data to the silica data and seeing if we can find a correlation there. And then we also did some experiments with sediments that were collected on the cruise, and we were using silica 32 as a tracer to try to understand the rate of silica uptake or the silica sequestration. So moving from the from like the soluble phase to the particulate phase. And I'm also looking to do some laboratory experiments. So we will be looking at specific groups of microbes. So the first group that we are interested in is Geobacter, because it is an iron-reducing genus of microbes, and reduced iron is a really important substrate for a lot of reverse weathering reactions, which are part of authigenic clay formation. So we think that because Geobacter is producing this reduced iron, that maybe it is accelerating authigenic clay formation. And the second microbe that we're looking at is Desulfobacter. So these bacteria, they produce hydrogen sulfide as one of their metabolic byproducts, and hydrogen sulfide can react with iron and form iron sulfide minerals. And so we think that maybe there might be some connection there with reverse weathering reactions and orthogenic clay formation because maybe they're reducing the availability of iron. And this is just a little overview of the experiments and how we're planning on doing them. And so it's a 5-day time series experiment. So we're looking at different time points. We're using silica 32 as a tracer so we can see how much of that silica is being sequestered in the particulate phase. And we will have treatments with both of our different microbial cultures. And in the lab experiments, we are creating a mock sediment slurry. So we'll be using a reference clay and some clean diatoms to try to replicate what we would see in the northern Gulf of Mexico, but we'll be able to have a control that is sterile without microbes. And this is some of our preliminary data from the field experiments that we did with the field sediment. And so these graphs are showing the data. We had treatments with microbicide and treatments without microbicide. So this is our treatments with the biologically active microbes. So, and this is showing you how much silica is being released from the sediment that was added to these sediment slurries into the solution. And then this is our treatments with microbicide. So as you can see, it appears that the treatments with microbicide are releasing more silica from the sediments. So the microbes appear to be causing less silica to dissolve.
All right, thank you. So we've kind of taken you on a little bit of a wild journey. If you came in halfway and you saw a bacteria slide or you saw all this other stuff, we do a lot of weird things in here, but they're all kind of under these central umbrellas of really trying to understand how an element moves through the not only the ocean, but through the sediment and the surface of the globe. Conclusions from some of the studies that we showed you today. Diatoms are important marine systems, but the silicon requirement, it really forces this kind of extra physiological constraint that changes how they operate in an ecosystem. So Antonia's data show this, but master student Kayla Barnett, she's also looking at these types of dynamics locally, and it's important. And what's interesting is you might ask yourself the evolutionary question, why on earth would any organism try to like willingly put themselves into limitation? Well, we actually have published something pretty recently. Our February speaker, Kim Tamatricon, led this, but it actually might benefit them because when diatoms are limited by silicon, their organic matter is still good. They're just kind of growing a little bit slower. But if you're limited by nitrogen or phosphorus, you're going to reduce your enzymes, you're going to reduce maybe your lipids, and you're going to make your organic matter kind of, you know, a little bit out of whack. Silicon limitation might actually be a good thing evolutionarily because it doesn't hurt them the way other types of limitation does. So it's kind of an evolving concept that we're examining. Diatom silicon requirement, because they have this and they're kind of unique, this gives us a really unique way to follow one group through an environment. And more than that, we can follow the group not only through the environment, but we can follow the fate of that material into different sediments. So what Lacey was talking about is in that Southern Ocean, you saw that big transition. The Southern Ocean, because of the Antarctic circumpolar current, the Southern Ocean is the great silicon sink in the ocean. The spillhouse projection shows the Southern Ocean very centrally because it connects all other sectors of the ocean. But it's basically kind of like a giant sink for silicon, and the concentrations there are far exceed pretty much everywhere else in the environment. And then we can also think about, well, what happens when that material gets down to the sediments? Because if that silica can stay intact and that carbon stays in there, that's a net sink of carbon dioxide, right? And if you can bury that, that brings it out of the atmosphere for potentially millions of years. But if it dissolves, then you can release that organic matter to respiration. You could potentially put that silicon back into the water from our diatom activity. But if it stays in there, and Priya's data and also Kendall Breland, who's in our lab, is a PhD student on this project as well, they're showing that maybe microbes are helping kind of like grab that back. And when they do, it kind of reacts this second pathway, this reverse weathering pathway, which means that that carbon dioxide is now being produced from the formation of these new clays in the sediments from that silicon. And so it's this kind of big cycle that we're thinking about. And it's something that I said, you just kind of wandered, we just kind of wandered into because of one of Dr. Carmichael's students coming to my lab in 2013, just asking about sediments. So looking ahead for us, well, you know, we work in a lot of different places, but our February speaker, Dr. Tamatrikon at Rutgers, and I have a pending proposal to look and see what happens with viruses and diatoms and how the viruses change this. And we want to go to look in the open ocean because we have an understanding of what viruses do in the coastal ocean where they're really abundant, but diatoms are much less abundant in the open ocean. And for a virus, that is a problem because they're not a big target. And so she, through her lab, we kind of published this paper, which NSF put on the website, which was pretty cool. Algae killing viruses spur nutrient recycling in the ocean. We want to take this further. We want to go to Bermuda with it. Oops, sorry. And then we also want to think more about how do we integrate these mass spec technologies into it. So that's the end. We got a lot of acknowledgements because, well, we covered a lot of ground, but clearly we got to acknowledge National Science Foundation. You know, Antonia's work also was funded by the Helmholtz Young Investigator Group in Germany. We have lots of collaborators. We go out to sea a lot. And then also we have, you know, a series of kind of team members that have kind of set the stage for the research we're doing right now. But with that, I'd just like to say thank you. And I think we have time for questions. And I'll invite the other speakers to come up so you guys can fire at them as well. All right. Dr. Smee. Thank you. Antonia, you want to take a stab at this? So the question is, why is silicon limiting in the Arctic?
So, I mean, at the end of the day, it's probably a question of both. So nitrate is definitely limiting at some point. And the nitrate concentrations that we saw in our cruise were all very low. My question was more like, so the general assumption of that is normally that people are like, okay, so nitrate limits primary production and then we can ignore everything else. But what we can see is that there's a specific effect of the silicon concentration on diatom productivity aside from whether there's nitrate or not. So kind of the assumption is less that silicon is more relevant in limiting primary productivity. That is probably still nitrate, but that silicon probably influences how the community shifts and when the community shifts towards kind of away from diatoms. Yeah, not sure if that answers your question.
Yeah, so the question was, is there another example of where you have a lot of nitrogen, maybe from eutrophication, and silicon might become limiting? This is actually something this was one of the justifications for the first project we did in the Gulf. Mississippi River plume and the whole watershed is being fertilized excessively by nitrogen and phosphorus, and it wasn't always that way. And there's also been, on the Mississippi, in the watershed, we have changed how water flows, especially with dams. And dams lead to reservoirs and retention of particles because instead of a diatom shell floating downstream and dissolving, it hits a big wall and sinks. And so what we've seen in the Mississippi River over time is you've seen an increase in nitrogen and phosphorus, but a decline in silicon. And so Gene Turner at LSU and Nancy Rabelais have looked a lot at, well, what's happened in the sediments, and their hypothesis, which I think we've given a lot of kind of credence to with our publications in the last couple of years, is that eutrophication has shifted the system away from diatoms. And this is also similar to what you see in lakes. If you ever look at your dish soap and it says phosphate free, it's because phosphate in the Great Lakes led to a monster diatom bloom. Those things sank all the way to the bottom of Lake Michigan, and they saw that the sediments were just chock full of diatoms because of this eutrophication. And that led to actual, mitigation and regulation because of it. In the North Atlantic, it's a weird site because the silicon's really low unless you get that overflow stuff from like the bearing, and the nitrogen usually will start the season a little bit higher than the silicon, which is a unique thing in the ocean. All right. Any questions on Zoom? Nope. All right. Well, can you give a hand to these folks?